In this video, we are going to be looking at the stages of meiosis and mitosis. The first image is a diagram of the entire cell division process. Shown at the top is the mitotic division section of the cycle, which includes all the steps of mitosis. To the right is G1, which involves growth and normal metabolic action of the cell, and is also the step at which the cell decides whether or not it is going to divide. At the bottom is the S phase, or synthesis, which involves the replication of genetic material inside the cell. Finally is G2, which is growth and preparation for mitosis. Not pictured is cytokinesis, which happens after the M phase. Shown here is a cell and interphase of mitotic division. It is the longest process in cell division and the most abundantly seen. Mitotic division is the source of growth and repair in organisms. Cells replicate themselves identically to replace damaged cells or to grow overall. This process is essential, and without it, macro-sized species would never have evolved. Mitosis results in two daughter cells, each with a diploid set of chromosomes. Shown first is interphase. By this time, DNA is replicated, but not condensed into chromosomes. They remain as loosely coiled chromatin. Next is prophase. Now the DNA is condensed to form chromosomes. The nuclear membrane is no longer visible, and the spindle apparatus has begun to form. The next two steps are metaphase and anaphase. In metaphase, the spindle fibers have attached themselves to the centromeres of the sister chromosomes, and the chromosomes align at the equatorial plate. In anaphase, the spindle fibers shorten, and the centromere splits. The sister chromatids are then pulled towards their respective poles. Finally is telophase. The chromosomes reach the poles of their respective end, the nuclear envelopes begin to reform, and the spindle fibers slowly disintegrate. Cytokinesis then follows, and the final results of mitosis are two daughter cells with diploid sets of chromosomes. Meiosis is a different, but just as important process of cell division. It is the process of cell division that creates genetic diversity. Meiosis matters because it is a central part of evolutionary theory, allowing for genetic diversity, which in turn gives the opportunity for random beneficial adaptations and mutations to the environment, i.e. a better level of fitness. Without meiosis, species would be genetically identical to their parents, and there would be very little diversity. Without it, evolutionary processes would rely solely on random genetic mutations. The first step to meiosis is prophase 1. There are two end parent cells with the genetic material that has already been duplicated. Synapsis occurs, where each pair of chromatids joins its homologous pair to form tetrads. During this, recombination is possible, which is random crossing over bits of genetic info in the tetrads that creates genetic diversity. The mitotic spindle also begins to form. Next is betaphase 1, in which the tetrads line up on the equator of the cell, during which time the spindle fibers attach to the centromeres of each tetrad. The next step is anaphase 1, in which non-disjunction occurs and the two chromosomes of each tetrad separate, beginning to move towards the opposite poles of the cell via pulling of the spindle. Sister chromatids remain attached at their centromeres. The final step in meiosis 1 is telophase 1, in which homologous chromosome pairs complete their movement to the poles. A haploid set of chromosomes is at each pole, a nuclear envelope reforms around each set of chromosomes, and the spindle disappears. Meanwhile, cytokinesis begins. Cleavage reforms at the two new genetically unique haploid cells, and thus completing meiosis 1. Next comes meiosis 2. Prophase 2 has no new chromosome replication. The centrioles duplicate and separate into two centrosomes, one for each chromatin. The nuclear envelope breaks down and the spindle apparatus begins to form. In metaphase 2, each daughter cell finishes formation of the spindle apparatus. Single chromosomes are aligned on the metaphase plate, as opposed to homologous pairs, and microtubules connect to each connector core. Anaphase 2 is when the centromeres separate and the two chromatids of each chromosome move towards the opposite poles of the spindle. Finally, there is telophase 2, in which the nuclear envelope forms around each set of chromosomes. This is followed by cytokinesis, and eventually the production of four daughter cells with haploid sets of chromosomes. The final result is a set of four daughter cells each with haploid sets of chromosomes that are all genetically unique.
On that day my son was born.